All right, so we're now going to jump into part two of our Protists lecture. And again, remember, this is a moving target because of new information, new research, molecular data especially is providing us with lots and lots of new information and causing us to rethink the classification. So if you reflect back on classification, when we did that lecture, we talked about how you look for traits and you group things together based on features and characteristics. And we use that idea of parsimony. Simplest explanation is generally the most accurate. Well, that's what we're doing here. As we reclassify protists, we keep moving them around and reorganizing because of parsimony, because of this seems to make the most sense. This is the simplest explanation now that we have this new piece of information available. All right, so here is the visual picture I want you guys to think about as we work through this lecture. And honestly, this is a really good diagram to draw out. Don't just print it or take a screenshot because then it's, okay, minimal effort to do that. But draw it out. That way, as you're drawing it or you're writing it out, you're investing more time and energy into it, and it's more likely to stick in your head and be there when it's time to do this type of stuff on the exam versus, oh, I just saw the picture and printed the picture. So what we're going to do is we're going to take a look at the protists and look at them in their supergroups. So these check marks represent the 6 supergroups that are currently being used to organize the protists, well, and everything else. If you notice, if you can see it, you got land plants here, you have fungi here, you have animals here. Well, those are kingdoms. They're still established as kingdoms of life, but now they're being housed under these different supergroups. So kingdom Animalia, kingdom fungi, plus the quinoflagellates all go under supergroup Obstacanta. The land plants, kingdom Plantae, is now grouped with chlorophytes, the charophytes, and then rhodophyta under supergroup Archaeoplastida. So, okay, so again, we're going to go through this. What I want you to be able to pull out of this is not so much all the little details of the organization, but what feature or features, sometimes there's a couple, are required to place an organism in a particular supergroup. So what do you have to be a member of Chromoalveolata? What about Rosaria? What do you have to go into that supergroup, and so on for each of these supergroups? Within a couple of them, we're going to drop down to these subcategories. So I'm not going to call these kingdoms or phylums or anything like that at this level. That's still being worked out. But if you're a Chromoalveolata, what then allows you to go down to Alveolata or Straminopilla? What are the key features? So we're going to work through this. This is going to be a long lecture collectively. This is why I build in multiple days for this one. Don't try to do this all in one shot. It's going to take a bunch of time, and I'm going to walk you guys through it and show you how to put the information down so that way it's hopefully easier to process. Okay, so I want you to think about the dichotomous key. We're basically going to build this like a dichotomous key. Give me 1 second. Let me do something here just to show you what I mean. Let me get a... Okay, so the idea behind the dichotomous key, and I'll walk through this with you as we go. We're going to start here. This is supposed to be supergroup. What are the key features? And then if you go down this supergroup, maybe you go into this group or this group, what feature do you have to go this way? If you go this direction, what feature do you need over here? Once you're in this category, maybe you go in one of 3 different directions. Okay, what feature is required for this line. What about this line? What about this line? All right, so that's the idea of the dichotomous key. You guys walk through those steps, and when you hit a certain feature, it caused some of the organisms to go one direction in classification and some to go a different direction. It's exactly how we're going to approach this. Okay, so hopefully it's a little familiar now that we've worked through dichotomous keys, and you have hopefully a little bit of familiarity with it. So, all right, so just for the visual people in the group here, this is a spectrum of protists. There's all sorts of craziness out there. We saw a little in that video. You have these things called amoebas, single-celled. They're kind of like an animal. They eat stuff. They digest it. They poop out the waste. Well, it diffuses out. They don't have a digestive system, but it's a single-celled organism. So is it an animal? Not really. What about these? Volvox. These are photosynthesizers. Those little green dots run photosynthesis. They're colonial cells. They all group together, but they don't form a multicellular organism like a plant. There are multicellular protists. We'll get into some of those in a bit, okay? So there's just this amazing diversity here. And the crazy part about protists, I am almost willing to bet you guys 20 points. You had a protist in your life today. In some form or some fashion, there are different ways. I guarantee protists are part of your life. So, and I'll be pointing that out as we walk our way through the different groups and talk about them and what they do. I want you guys to remember some examples of protists that fit in these different categories, and then we will discuss our uses. There's good ones, there's bad ones, there's ones that were like, what's the big deal? It's a protist. So, all right, so away we go. Our first supergroup to explore is supergroup Excavata. So I'm going to kind of move around within that picture, but I want to start here with Excavata. This is supergroup number 1 for us. So that's supposed to be a supergroup. All right, so actually let me get rid of this, and instead of the hashtag, I want to put a big red star. Oh, too much. What makes somebody a member of Excavata? There's our big red star. This is your subtle hint. That's supposed to be a star. Work with me, guys. This is your subtle hint. Make sure you know this. This is going to be a key piece of information for you to remember. Yes, it'll be on your exam. All right. So to be a member of Excavata, you're a protist. You're these goofy, weird things. But the big defining feature is that the protists that are under Excavata all have a groove on the side of their body. So when we look at them physically, they all have this groove or indentation that runs the length of their body. That's it. Wow, that's real exciting, right? But that's all it takes to be Excavata. When we get into Amoebozoa and Archaeoplastida and Rosaria and so on, there will be different features. All right, so again, we'll go through all this. Let's just work through Excavata first. All right, now within Excavata, we have 3 subgroups. The first one are the diplomonads. The second subgroup are the parabasalids. And the third subgroup will be the euglenozoans. So what we're going to do is we're going to work through each of the subgroups, talk about key features and characteristics of those subgroups. Again, pros and cons, why we like some of them, why we really don't like others, what do they do, and what are their defining features. What makes an organism a diplomonad versus a parabasalid? But you have to be an Excavata first. So again, what's your first criteria? And then what's the secondary criteria to go into this category versus that one versus that one. So, and I want you guys to remember the dichotomous key structure and concept. That is so important to making your life easy with this particular unit, at least the Protists. All right, so first group of Excavators to talk about are the Diplomads. So, and I'm just going to put it up here. Remember, these are part of supergroup X, and I'm writing it because it looks horrible, but you'll hopefully remember it more versus if it was just typed out and looked all neat and clean. So, all right, supergroup Excavata. So, Diplomads. What do we know about them? Well, they're all single-celled. That's a unifying feature here within this group. They move with a flagella. So there's that little whip-like tail, that flagella they use to propel themselves around. And they will have 2 nuclei. And then when it comes to their power source, they have what are known as degenerate mitochondria. So the mitochondria are not completely structurally functional and structurally the same as what we see in most other eukaryotes. So they're a little bit of a different format or kind of style for their mitochondria. So you take those 4 characteristics, put them together, you have a diplomonad. Along with a groove on the side of the body, you now have an excavata, a diplomonad that sits in the excavata. All right, so other organisms will be single-celled, but they're not going to have the other 3 characteristics. Other organisms might have 2 nuclei, but they're not going to have all 4 of those characteristics. So that's the key here. Those 4 combined make you a diplomad. To be excavata, you have to have, and I'm not going to tell you, I want you guys to hopefully remember, because I've said it multiple times, you're like, oh yeah, I'm remembering the excavata feature. Okay? An example, let me give you guys an example of a diplomonad. Let me find, I got to do it in italics because it's a scientific name. Giardia. So this is a parasitic diplomonad. This is why you don't go and drink water out of the river or out of the stream or out of the lake, because if there are Giardia in it, then they are intestinal parasites, and that does not do us any favors. That's not going to make you have a good day. Giardia can be transmitted by drinking infected water. Beavers often harbor Giardia populations, and it can lead to some health problems for people, again, if you get infected. So if you're out hiking, camping, fishing, etc., be careful. Filter the water, boil it, clean it. You boil it, you destroy if there is Giardia in it, you destroy the diplomonad. Sadly, you go around Illinois and the United States and test bodies of water, you're going to find Giardia in it. The majority of aquatic ecosystems will have Giardia. It's just what level of Giardia is present. Is that enough for it to lead to a problem with us if we come into contact with it? So it's a matter of if we drank that water, we're swimming and some got in our mouth, did we get enough that it's actually going to become problematic? So, okay, so I'm going to stop this one here, and that way you guys could break, go do something else, come back, or click on the link and we'll move into the parabosolids in the next lecture.